Good afternoon, everybody. Myself, Dr. Revati BR, Department of Biochemistry. Our topic is collagen. We know what is collagen. Collagen is the major structural protein in the connective tissue. About 25 to 30% of the total weight of protein in the body is collagen. And it is the major fibrous element of the tissue like bone, teeth, tendons and cartilage, blood vessels. Coming to the type of the collagen. Collagen can be classified as its distribution: type 1, type 2, type 3, type 4, type 8, 9 and 12. And the main classifications are fibril forming, network forming and fibril associated. The fibril forming collagen consists of type 1, type 2 and type 3. Type 1 is seen in skin, bone, tendon, blood vessels, cornea. And type 2 is seen in cartilage, intervertebral disc and vitreous body. And type 3 is seen in blood vessels, skin and muscles. Then network forming consists of type 4 and type 8. The type 4 is seen in basement membrane and type 8 is seen in corneal and vascular endothelium. Then the third type is fibril associated collagen that consists of type 9 and type 12. The type 9 is seen in cartilage and type 12 is seen in tendon, ligaments and some other tissues. Is it okay? Then the elaboration is same. The fibril forming collagen. Fibril forming collagen consists of type 1, type 2, type 3. It is a rope-like structure and in microscope we can view in it's a bending like pattern. In type 1 has high tensile strength and it is a supportive elements. Type 2 is seen in cartilaginous structure and type 3 is seen mainly in blood vessels. The network forming collagen seen in basement membrane, it consists of type 4 and type 8. The fibril associated collagen consists of type 9 and type 12. It is seen in extracellular matrix. Coming to the functions of the collagen. These are the main functions of the collagen. And it has a great tensile strength and it serves to hold the cells together and it involves the proper alignment of the cells in blood vessels. If the collagen is exposed, the platelet adhere and it helps to the formation of thrombus, that is, it helps to the clotting system in our body. The collagens are seen in cartilages, bones, hair, connective tissue and nails. Then coming to the structure of the collagen. The basic structure of the collagen is tropocollagen. This is the tropocollagen. It has molecular weight 285 kilodalton and this made up of 3 polypeptide chain. These are the 3 polypeptide chains which held together by a hydrogen bond. They are twisted around each other like triple helix structure. This is the triple helix structure, that is the rope-like structure and held together by hydrogen bond. Each polypeptide chain consists of collagen about has 1000 amino acids residues. Every third amino acid is a repetitive, that is, it is represented as glycine XY, then again glycine XY. The X and Y are the other amino acids. These are the structure. It is helical structure that is twisted around as a triple helix structure and bonded by hydrogen bond. Coming to the elaboration of this structure, triple helix structure. Collagen is a fibrous protein, has elongated triple helix structure and stabilized by interchain hydrogen bond. Then hydroxyproline and hydroxylysine residue. The collagen contains hydroxyproline and hydroxylysine, but it's not present in other protein. Then coming to other property is glycosylation. What is glycosylation? The hydroxyl group of hydroxylysine residue of collagen enzymatically glycosylated. That is, the glucose and galactose sequentially attached to the polypeptide chain. It occurs before the triple helix structure formation. Then coming to the synthesis of the collagen. The collagen is synthesized by fibroblast intracellularly as a large precursor called procollagen. After the synthesis, then it's secreted into the outside of the cell. The extracellular procollagen is cleaved by a specific peptidase to form tropocollagen. That is the basic form of collagen. These are the synthesis of steps in the synthesis of collagen. First is undergo transcription, then translation, then prolyl and lysyl residues are formed. These prolyl and lysyl residues undergo hydroxylation, then it forms hydroxylysyl residue. Then it again undergo glycosylation, form procollagen. The procollagen then chain association and disulfide bond is formed in the procollagen. Then it undergo triple helix structure formation that is secreted into the extracellular matrix as procollagen. Then there is conversion of procollagen into collagen. Then cross-link formation is happen. See, up to the glycosylation, it is seen in intracellular. The other processes are seen in extracellular. This is the maturation of procollagen into collagen. That is, the procollagen is synthesized in the intracellular, that is hydroxylation and glycosylation then secreted into the cell as procollagen. Then the triple stranded helix structure is formed in extracellularly. It is cleaved by the peptidase. Then tropocollagen is formed. Then tropocollagen assembled into the collagen fibrils with a quarter staggered arrangement. This is the overall process that takes place in the formation of collagen, that is intracellular alternations and extracellular alternation. What happens in the intracellular alternation? There is hydroxylation of proline and lysine residue, then glycosylation of some hydroxylysine residue, and there is formation of triple helix. Then in extracellularly there is cleavage of the portion at both the carboxy and amino terminal end. Then the formation of quarter staggered alignment and the oxidative deamination of the epsilon amino group of lysine and hydroxylysine residue and formation of intra and interchain crosslinks. Coming to the degradation of the collagen. Then the proteolytic activity of the collagenase enzyme, which is produced by the Clostridium histolyticum pathogen, which also causes for the gas gangrene, which helps to the degradation of the collagen. The degradation seen in bones, cartilage reabsorption, osteoporosis, tumor metastasis, postpartum involution of the uterus, Tackus disease, rickets, and osteoarthritis. Is it okay? Then coming to the collagenopathies. First of all, we are going to discuss about the brittle bone disease, the famous brittle bone disease. It is otherwise called osteogenesis imperfecta. What happening here? It is a inherited dominant trait, that is a mutation occurs in the type 1 collagen, which results in replacement of the single glycine residue by cysteine. These changes disrupt the triple helix structure, hence the polypeptide became excessively glycosylated and hydroxylated, then unfolding of the helix takes place and fibrillar array cannot be formed, and this result brittle bone. These are the symptoms seen in osteogenesis imperfecta: the curved spine and the pale blue sclera, and it is associated with dentinogenesis and impaired functions of cardiovascular system and lungs, etc. Then coming to the next collagenopathy, Ehlers-Danlos syndrome. That is, its condition results from the defective in type 3 collagen, that is defect in the lysyl and lysyl oxidase or lysyl hydroxylase. It is characterized by the weakening of the collagen, that is loose skin, hypermobile and lax joints. The picture represents the lax joints, hypermobile and loose skins. Then coming to the next one, Marfan syndrome. It is an inherited autosomal dominant manner defect in the gene coding for connective tissue protein called fibril 1, that is defect in the fibril 1. It is also a component of microfibril. It characterized by ectopia lentis and hyperextension of the joints, arachnodactyl, long digits, and aortic aneurysm are its manifestations. Then coming to Menkes disease, it is due to the deficiency of the copper, which result in the defective of lysyl oxidase, which reduces the cross-linking of the collagen. And last one is deficiency of ascorbic acid. It is characterized by the defective or hydroxylation of collagen, that is the collagen is formed is very weak and leading to the fragility of the blood vessels and poor wound healing. Thank you.